My name is John Long, and I'm a desperate man. Maybe I should say I'm a desperate professor. I teach biology, cognitive science. I do research in those areas, and there is stuff that you can't normally do. There are questions you can ask that are really hard to answer. So I want to talk to you today about trying to get new methods using robots to study biology. And that's what this book is about. If you happen to have taken a look at it, you'll know some of the things that I'm planning to talk about today. So I want to know, how did vertebrates evolve? You know, just a little question, right? It's our subphylum. Hey, how did they evolve? Well, it happened 500 years ago. So how do we get at the answers that have to do with the process of biological evolution? And this is a beautiful illustration of what we think fish might have been doing 400 million years ago. And the first vertebrates were fish. So really we're talking about the evolution of fish, and they were big, fast, and hungry. And one of the things about them is they came in all kinds of forms. Most of them didn't have jaws initially. And they evolved, they brought with them into life, these vertebrates, a couple of really cool features that we hadn't seen in this configuration before in life. And those features are a new kind of skeleton, an internal skeleton that has a vertebral column. You guys all have one of these. I'm going to call it a backbone today. And a special kind of head. And you can see it in this sort of ugly form right here in front of you. Or you can think about a beautiful head of a fish. And what it's got that's special is a tripartite sensory system. That is nose, 2 nostrils, 2 eyes, and 2 ears. That's the new vertebrate head to go along with the new vertebrate internal skeleton. So I want to know, as a biologist, I'm desperate to know, what were the evolutionary processes that generated these new innovations in life? So part of what we need to do is think about, well, what was going on before vertebrates were around? These are beautiful fossils from British Columbia, from the Burgess Shale. And these are wonderful invertebrates. So before there were vertebrates, there were all the rest of animals, and they were getting along fine without us, right? So here's a marella, which is a kind of crustacean-like critter. Here's a velvet worm, famously upside down in this picture. They, we think, moved around using flexible appendages, and these guys had a hard external skeleton that you all recognize by the term shellfish, okay? And here's how one artist conceptualizes Opabinia from this same group moving around. Talk about a strange animal, right? With this sort of mouth, jaw thing. You don't know what's going on here. I don't quite understand the background of this picture. It looks like they're worshiping sun rays. Okay, they were obviously at the top of the food chain there. But really, when vertebrates were first evolving, invertebrates were the top predators. The New York State fossil, anybody know what that is? No, it's not a trilobite. Back there, yes. Eurypterid. What's your name? Michael, way to go, Eurypterid. Giant sea scorpions, they could be 6 feet tall, and they're crustaceans, right? Totally awesome killer invertebrates. Okay, so keep that in mind. Now, the problem is we have all these great fossils, right? But fossils only tell part of the story. Here are some beautiful fossils. Believe me, they're beautiful. These may look like smudges to you, but here's a paleontologist's drawing of these smudges, and you can see the nasal cavities, the eyes, and the ears here. Here's another headshot. We know these guys are vertebrate. Coolest thing ever, 500 million years old. Next coolest thing, this is soft tissue. Soft tissue doesn't normally fossilize. So we have this great record of these fossils from China that have come out in the past decade. And these guys are the first fish. They're 530 million years old, and they advanced the age of complete vertebrates that we had in the fossil record by 100 million years. Totally awesome fossils. Now right here you can see some little scratches, it looks like, in the rock. Those turn out to be muscle segments. You may recognize, if you've ever eaten fish, the muscle segments that fish have. And there's also here little blobs of cartilage, or what we think was cartilage, that are protovertebrae. First early experiments in laying down on a cartilaginous rod that's called the notochord the things that will eventually become bone. All right, so fossils are awesome, but dead fossils tell no tales, right? And there are no witnesses when there are fossils. So how do we get at the behavior that these animals have? Behavior doesn't fossilize, right? So we have to do other things. And there are two major approaches to this. One is to study living fishes and try to guesstimate, based on a diversity of fish forms, what extinct forms may have been doing behavior-wise. And the other method is to employ model making, right? Make stuff. And so I do both. I study living fishes and I create living robots. No, that's not going to work, is it? And I create autonomous robots. They're not living, but they're behaving. So that's what I'm going to talk to you about today, is how we do this process. And keep in mind that the reasoning that we have as we go forward is much like a crime scene investigation. And when you're in a crime scene, you have clues, physical evidence, and that physical evidence allows you to address the question of what happened. Fossils are great for that. We can see patterns, we can see the new occurrences of really cool animals like the first vertebrates, Haiku-ichthyi. But it's really hard to answer that question of how did it happen. So I would like to propose a new kind of CSI in their franchise, right? It seems to go on and on. What about CSI evolution? Wouldn't that be cool? So here are a bunch of people investigating the evolutionary crime scene. And so they're looking for clues of what happened. Okay, and in this particular dig that they were doing, was from the Devonian, which is in the age of fishes. And again, we're still left asking the question, though, of how did the evolution occur? So we start with the fossils, and that gives us some very useful information. So how do you make robots? In particular, I'm going to talk to you about a special kind of robot called a biorobot. These are robots that are designed on purpose to mimic certain aspects of living animals. Okay? And a big deal is that your biorobotic model has to represent, in some form, the biological target. Now, what's really important about this is, even though we can build a model, and here's a Tadro, which is one of our biorobotic models that I'll tell you more about in just a minute, and we can reference it to a 400 million year old fish, Drepanaspis, there is some implicit logic in what we're doing that you might disagree with, or you might think that's okay. I will tell you it's controversial. Right? I have good colleagues who, when I tell them, yeah, I'm working on robots to try to study ancient fishes, and they're like, what do robots have to do with biology? And I say, you know what? If you think that you can use a computer model to learn anything about the real world, then a robot, if it's properly designed, is a model in the same sense. So let's talk just a little bit about the logic of biorobotic models in biology. So first of all, oops, I have to get rid of your water there, sorry. First of all, the model represents the target. Very obvious, right? But believe it or not, there are people who build lifelike robots, and that's not a concern. That's not biology then, that's artificial life. It's a beautiful field that does that. Okay? Secondly, the model is a simplification of the target. It has been said that the world is its own best model. Rodney Brooks, a famous roboticist, has said this. Norbert Weiner, the father of cybernetics, talks about the best model of a cat is a cat, right? And these are all criticisms of a kind of modeling. And so we build models that are simplifications so that we can understand them. I work with a lot of engineers. I'm not an engineer myself. And engineers hate building simple stuff. They have gone through school, they have been trained, they've been given the tools to do stuff that's complicated, to make complicated things. So we use an approach, actually, that comes out of engineering. It's kind of a counter approach called the KISS principle, right? Many of you probably do that yourself when you make stuff. Keep it simple, stupid. When you build something simply, you understand that first, and then you can incrementally add complexity as you go forward. Okay, so also we pay attention to the functional mechanisms that are operating, things like brains and sensory systems, and the structure, the anatomy of our system. And finally, when we do all this, what we claim is that when we test a biorobotic model, we're learning something about the biological model. And the problem is with fossils, we can't go back and compare our model to the living fossils. So this is in the world of how possibly things might have happened. We can never claim that we know exactly what the answer is, but we can circumscribe the possible and eliminate the less likely when we do this kind of work. So that's where we're going with biologically. Let's take a closer look at our models now. So I introduced Prero to you, or Tadro, and this is a particular kind of Tadro called Prero. World of robotics, right? Everybody needs stupid robot names. It's much of the fun of robotics. And so here this stands for tadpole robot. Prero is prey robot. We're very creative nerds. Okay? We're very proud of our stupid names. And so what we have in Tadro is a couple of those very important sense organs that we talked about vertebrates having with their new heads. Here are paired eyes, this thing called a lateral line. Michael, you still there? So can you tell me what a lateral line is? Oh, dude, I mean, how much do I have to pay you? That was perfect. So Michael is saying lateral line is something on the side of the fish. It senses movement in the water. That's perfect. How does that relate to the nose, eyes, ears? It turns out it's an inside-out ear. Or the way you can think about it is our inner ear that has little hair cells in it encapsulated with a piece of the ocean is an outside-in lateral line. We carry around our heritage stuffed in our inner ear. Very cool stuff. Love the evolution. You know, it's building on top of what you've already got. Okay, so we take two of those things. We have a brain that we're going to, in a very simple way, try to replicate. And we're going to try to evolve the tail. And we're going to pay particular attention when we do that to building a biomimetic backbone with variable numbers of vertebrae. And we need variable numbers because we're going to apply artificial selection and let evolution, which is an automatic design process, right, hands-off design process, tell us what the best solutions are, right? We don't know ahead of time what's going to happen when we start evolving robots. So, I can tell you, robots will not take over the world. Evolving robots will not take over the world. I can tell you that. So, what we've got here is those eyes turn into photoresistors, and then the lateral line, we use an infrared proximity detector to estimate that ability that Michael was talking about there. And I've mentioned to you the biomimetic vertebral column. So here's a diagram of our students at Vassar College making these biomimetic backbones. We start with some molds, which are simple cylinders, and after we make what's the equivalent of a notochord, which is a flexible and wet, okay, biology is wet, it's a flexible and wet structure, we then slide on and glue what are called ring vertebrae to it. So that's a hydrogel of 10% gelatin. You can't eat this stuff, okay, because we cross-link it turns out, with a chemical to keep it stable over time. And then we add variable numbers of ring vertebrae out of Delrin. We slide them on. These are just holders on the end. And then we can vary the number from 0 to 11. We pay a lot of attention to what we know about the vertebral columns of sharks. Part of what we do in my laboratory are biomechanical tests. So we actually take the vertebral columns of sharks and subject them to engineering tests. And we make sure that our biomimetic vertebral columns match up in terms of the mechanical property, the stiffness, the elastic energy storage. So we're doing the best we can by working on living species to guesstimate what may have been going on in that ancient fish, Drepanaspis. All right? So that's our biomimetic backbone. Out of this, though, so now we have some anatomy. We need robots that are going to behave. And we don't want to remote control them. We want to have them on their own make decisions about what they're going to do in the world. So the way you make behavior is not by remote control. So this is robot Madeline, and I don't know if you've heard about her before. She's our four-flippered robot built to mimic some of the plesiosaur behaviors that we see 60 to 90 million years ago. Here's robot Madeline now unplugged and in autonomous mode. So when you're in autonomous control, what we mean is that you have behavior. and the robot itself is in control. There's nobody behind the scene. I love the Asimo robot that the Japanese have built, that does this kind of thing, walks upstairs and stuff. The first time I saw one of these videos, I'm like, who's that guy back there behind? And there was somebody sort of remote controlling Asimo. And Asimo is a beautiful piece of mechanical engineering, but at least the Asimo I've seen is not autonomous. So you don't want anybody behind the curtain. So that's robot Madeline. And so how do we get autonomous behavior out of a Tadro? The trick is, when you do that, you can do this at home, Michael. The trick is to worry about sensors and their connection to motors. That's the trick of autonomy. So for example, if light hits one of the light sensors, then this behavior inside that we control with a microcontroller, like an Arduino, tells the animal to do its feeding behavior. And that's nothing more than turning and heading towards the light. At the same time, there may be a predator nearby that kicks off the IR proximity detector. And then the brain says, okay, well, you better flee, you better get out of the way. Sounds good, but now we have a conflict. Are you going to eat or are you going to be eaten? And that's where we actually now go into the neuroscience of how fish behave right when this happens. How do fish solve the conflict? They actually suppress that feeding behavior, which sure as heck makes sense evolutionarily, right? You could love chocolate donuts. I love chocolate donuts. It's really hard to get me to stop eating chocolate donuts if they are still in front of me, right? But I will run from the chocolate donuts if you put a gun to my head and threaten to kill me, right? So it's the same sort of thing here. We are built to preserve ourselves first and to feed later. So this sensory motor control of Tadro is based on what we know about real fish operating. Now, for those of you who care about this kind of stuff, this is interactive C code right here. Here's a bit of it that takes in the sensor readings and outputs a motor output in response to that. Okay? And again, the key here is a change in sensory input alters the motor output. So that's how you get autonomy out of these very simple kinds of robots. And here is now Prero in the flesh, as it were. And who wants to guess the name of our predator robot? Think, stupid nerds. Nerd names, excuse me. Stupid nerd names. Pardon, I'm sorry? I can't hear. Jaws? No, that's not bad. Not bad. And Terminator? Getting close. How about this? Tadiator. Hey? Tadro and Gladiator smushed together, a portmanteau. So we have Prero versus Tadiator. You can see the microcontroller here at the time we did this. This was an MIT handy board. Sorry, Arduino folks. We have the photoresistors and the IR proximity detectors. The only job of the Tadiator is to track down Prero. And it does it using what we think of as the equivalent of visual predation with an IR beacon. Okay, so this is Prero versus Tadiator. And now we can ask this question of how might vertebrae have evolved? But we can ask it by thinking, first of all, what does the fossil record tell us? Well, it tells us that the notochord came first. This is actually a diagram of a coelacanth, and you can see it looks like there's something missing right here. Well, what's missing are all those little bones that you can see in this beautiful X-ray by Tierney-Teese that we call the vertebral column. So when I put an arrow here like this, I'm not saying that coelacanths evolved into lookdowns, which is that species there. Okay? What I'm saying is that particular character of a notochord that came first in evolution, and it has evolved into a vertebral column, and actually many times. So it's a really cool thing going on with evolution. Now, so we've got that in mind, and then we can ask, okay, did the first vertebrates evolve vertebrae as an adaptation for improved feeding and fleeing? So why do we pick feeding and fleeing? So when we think about making a selection pressure, that is a criteria for survival and reproduction. So we talked about eating and avoiding being eaten. And look at this. Can you see what's happening here in this fish? Here's why we pick feeding and fleeing. If you get caught, you're done. So here is this fish, which was happy for a moment, until the big cliff fell on it and made it a fossil, right? So it got caught with this other fish in its mouth. And we know from living species of fish, again, that predation is an incredibly strong selection pressure. So we combine these two fundamental things to talk about what's going to happen in our evolving robots. So there's prey row, and there's tadiator chasing it. So light is food for prey row, and prey row is food for tadiator. So that's our setup here. And here is going to be tad rows playing the game of life. And let me turn on this video and tell you what's going to happen. So prey row is over to the left, tadiator's to the right, downsampled to 1 frame a second here. Oh, shoot, I can't use that laser pointer and also use the video. Apple, can you please do something about that? Okay, so I'm just going to have to talk to you about it. So prey row is just thinking about the light at this stage. Tadiator's on patrol. Now tadiator, right about this point, has picked up on prey row and is going to come around and try to get it right from its blind spot, which is behind it. Right? So prey row is happy. Chocolate donuts, found them, going to have them. Light is life. That's why we use it. Prey row doesn't know better now, but you can see tadiator's coming in for the kill, and prey row right about, you're going to see a big tail flip right about now, but too late, prey row is trying to get away, and so big points off in the game of life here for these robots. So this is a figure from Darwin's Devices, a different trial, right, where prey row is starting here, tadiator starting here, going second by second, and talking about all the interactions they have in a 3-minute trial. We run many of these trials on many different individuals of tad rows, all of whom have different numbers of vertebrae. And then what artificial selection is, which mimics natural selection, is picking the winners. Okay, so let's talk about that judging system. There are 2 things going on, remember. For feeding, we reward how fast you get to the the light source, and then how close you stay to the light source. For fleeing, what we reward is the distance from the predator, peak acceleration, your ability to get the heck out of dodge, hit that cheetah button and move, and then the number of escapes that you have. So we put those together, and from any generation we pick the best 3, and we allow them to reproduce. So here's what making evolution looks like. If you imagine for a minute, it turns out it's really hard to draw and show you different numbers of vertebrae up here and have them make sense. So imagine that the colors just represent different numbers of vertebrae. Okay, so keep track of the colors. So we have a population, and then we have the predators, tadiators, that are selecting by this judging criteria who gets to reproduce. So the survivors become the parents. They reproduce, and then we get the instructions for the second generation of those tad rows. Now, there's something missing from all of this, and that is that when you do evolution, what you need to think about when you compare what's going on with your generations, yes, there have been changes, and by the way, evolution is the change from generation to generation in the characteristics of the population, right? The big bummer for us is you can't evolve. I'm here to tell you, you cannot evolve. You can learn, you can develop. Michael, you can go on to be a roboticist and make specialist. You got it in you, I know. But it's populations that evolve, okay? And so when we think about evolution, we have to be careful. We're talking about change in the genetics of a population from generation to generation. That's what evolutionary biologists agree is how we know that something's evolving. So you got to have the genetics in there. And let's take a quick look at how we think about the genetics in our system. We do the selection that we just talked about, and then we mutate the genome of the robots. And what that means is, oh, I don't get the full 30 minutes, huh? Okay. And so we mutate the gene pool, and how we represent the genes, that's all done in software. And the software is genetic algorithms, and it's not much more than taking a number that represents a number of vertebrae and putting it through a random number generator that comes up with a new number on the other end. So that's what's represented here by the different hues. So the blue, let's say the top tad row, it gets to have 6 sets of genes in the gene pool. The second place tad row gets 4 sets of genes, and the third place tad row gets 2 sets of genes. So depending on whether you're gold, silver, or bronze, right? You get to represent your genome. We mutate that genome, and that's shown in the different hues. There were actually 3 different traits that we were evolving at the same time. B is the tail span. Zeta is actually the predator detection threshold. I'm only talking to you about vertebrae today. Okay, so then what we do is we throw those new genes together, and randomly we combine them into new kinds of offspring pairs. So this is going from, if you know your genetics, haploid gametes to diploid offspring. So these are the new genomes, and they give us the instructions for the next generation. So this is how evolving biorobots, this is how we do it, and we can witness now the game of life. I said there were no witnesses, right, happening 500 million years ago. So we've reanimated what we think is close to something like we're modeling that evolution of those first fish, and we get to watch. And you've already seen some of that in the video. So when we evolve those biorobots, here's one way that we track the evolution. So we have generation on the x-axis from 1 to 10, and then here's n, the number of vertebrae, and we start out our population at about 4.7 vertebrae, and that's the mean for the population. That's what this point represents. These error bars represent the standard deviation or something about the variation in our population. So they start out variable, right? Natural selection is choosing from different options. If everything is the same, there's no choice that natural selection makes, okay? So what happens over the first generation of selection? You can see we're actually running 2 different trials here. Both of them increase even more. Wow, this looks like pretty strong, what we call directional selection, and then they taper out. Okay, what's really cool about this, though, is the population is changing over generational time. We've determined what the genetics are, so we know there's a genetic basis of this, and this is evolution. And what's really cool is we can tie these structural changes, number of vertebrae, to a change in behavior. They get faster, and they accelerate better. So we have connected, by having an autonomous creature, if you will, modeled in front of us, we can make those connections between genetics, anatomy, physiology, behavior, and evolution. And that's really the power of this method. So when we ask, how do you make vertebrae? What we can say now, well, it's at least possible, according to our model, that the first vertebrates evolved vertebrae as an adaptation for feeding and fleeing. And this is a specific case of a general field that we've created called Evolutionary Biorobotics. And what you have to know to understand how adaptation works, which is the process of evolution by natural selection, are a number of things that we've talked about here. The great thing about modeling, and the modeling that we do, is that you actually can determine the genetics, the population structure, and the selection pressure, and then you measure these other aspects of your system. And what I've got for you here on the right-hand side is sort of a life cycle of evolutionary biorobotics, where we go from, we manufacture our new robots, we compete them, they get selected. This all happens in hardware. And then in software, that's this genetic stuff, from mutation, gametogenesis, to mating. So this is what we've been working about, and this is what is the story of Darwin's devices. So thank you very much. Any questions? We have time for a quick one. Yes, ma'am.
Can you use the model to study fish rather than vertebrates?
She's asking, can we use the model to study fish? Absolutely. And there are ways that you can, you're not restricted to trying to work in the fossils. You can actually try to make a model of a living fish as well. And there's a great laboratory at Harvard by George Lauder, and he's really working on biorobotic models to study living fishes. So yeah, that's a great point. Yep, we certainly can. One more quick question?
How do you determine hardware this make? What's your plan for that? Based on what happens in the interactions, how do you decide what you do with the hardware?
So he's asking, how do you decide what to do with the hardware and the enhancements to make? Because we start simple, it's always the question of what kind of complexity do we add in next, for example. So one thing that we'd love to do is have the predators evolve too. So it's really an evolutionary arms race. Right now the predators are not evolving. Another thing is we've got muscle equivalent just coming from one place. Having distributed muscle, there are some neat exhibits out there with muscle wire. So using muscle wire, which is hard to control, But that would be a neat thing to do, is put in a distributed force system. All right, thank you all.